Although there are deliberate attempts to harness the more beneficial side of viruses and convert them to useful purposes, viruses on the whole, of course, are rather horrible, and we can really need to look at them as renegade genetic sequences, and most of the time they're the cause of very serious disease. Now, what I want to do is to show you a couple of really important diseases in human history and contrast them and show you why some have been easier to control than others. The first one we want to discuss is a virus called smallpox. And its genetic material is double-stranded DNA, so just like the chromosomes in our bodies. And this caused horrible diseases. And we know that it infected humans for thousands of years. So here's a mummy from Egypt of Ramses V. He died about 1100 BC, and he died of smallpox. Here is a young man, 23 years old, who in 1923 was the last person on Earth to catch the smallpox virus. That was in Somalia, and it was in October 1977. Since then, smallpox has been eradicated from the world. But while it was around, smallpox killed 3.5 million Native Americans, and it killed around 100 million people worldwide in 3,000 years. It was one of the worst scourges in human history, but it's now gone. Now, while it was around, there were two major smallpox strains, one called variola major, which inflicted 25% mortality, and then there was variola minor that inflicted only 8% mortality. If you got the disease and you didn't die, you would then be immune for all the rest of your life. So survivors enjoyed lifetime immunity. Now, in the 10th century, the Chinese realized that if they could infect themselves with this minor form that only had the 8% mortality, that this would be much, much less harmful. And even better is if they took dried scabs from victims of variola minor and infected themselves, they would get fairly a mild case of a fairly mild strain of smallpox, and this would confer resistance against the vicious strain of variola major. In the late 1700s, a British physician, Edward Jenner, took cowpox, a virus that causes a similar disease in cattle, and he took it from his cow named Blossom, one of the most famous cows in Western history, and he used that to vaccinate a patient, Mr. James Phipps. And later, Jenner even vaccinated his own son with this cowpox, and it worked. Being exposed to cowpox provided perfect protection against smallpox. Now, cowpox was the mechanism of choice for a long time for vaccinating people. But the word vaccination actually comes from a similar virus to these pox viruses called vaccinia. And so here's variola, which is causing smallpox. There's a closely related virus that infects camels. And here's one that infects the cattle. And here's another one called vaccinia. And so the cowpox was eventually replaced by vaccinia, and this is where we get the term vaccination. And so you'd be exposed to this vaccinia virus that doesn't cause disease, but confers immunity. Now, remember, the immune system, when it's exposed to an antigen, mounts an immediate response with those plasma cells that produce the antibodies that go out and directly attack the viruses. But the memory cells are also activated at that point, and they're ready. They're ready in case they ever see that pathogen again. And so if you are vaccinated with vaccinia, you've got memory cells that have been produced, and years later, if you're exposed to something dangerous like variola that's so similar to the vaccinia, it's there and it recognizes it, and you'll be able to fight off the smallpox infection. So that's how vaccines actually work. So, eradicated by mass vaccination, it was possible to eradicate smallpox, because variola, the really vicious form, only infects people. Once it was eradicated in that last Somalia in 1977, it's gone. It doesn't exist anymore. But an important aspect of smallpox as a virus is that its genome is DNA. And as we're going to see soon enough, DNA as a mechanism of inheritance is quite stable. So the disease doesn't evolve as rapidly as some other viruses do. And so since these are all fairly similar, you can get cross-immunity. Exposure to camelpox is sufficiently similar that your immune system thinks when it sees variola, it's seeing camelpox again, and so it fights it off. So the stability of the DNA means it's a fairly stable target, and our immune system can be exposed to a similar virus, and we have memory cells that will protect us against the dangerous form for the rest of our lives. And variola, once it's gone from humans, is gone forever. Now, the reason for focusing on whether there are other hosts that can be infected by a particular pathogen is important when we start looking at some of these other diseases, and they're called multi-host pathogens. And around the world right now, in many parts of tropical Asia and Africa, there are new diseases that are appearing in our populations, and these newly emerging viruses are called zoonoses. And a zoonose is a disease that infects both humans and vertebrate animals. So, for example, SARS, which has caused some serious respiratory disease in Asia, actually lives mostly in the Chinese horseshoe bat, and people who live too close to Chinese horseshoe bats can get the disease. You can get rid of it in the humans, but you're not getting rid of the reservoir of the disease. So SARS will always be around as long as there's populations of horseshoe bats. Ebola is another multi-host pathogen. This is a really scary disease that can cause your body to hemorrhage completely and you violently explode with blood splattering everywhere. And the definitive host for Ebola seems to be in, again, bats, this time in fruit bats. And the disease travels from fruit bats into apes. And then in many parts of Africa where people are exposed to recently killed apes, they might get the disease from them. And so again, you can control the disease in humans, but there's a reservoir out there, and Ebola is likely to keep coming back and coming back as long as there's this reservoir of fruit bats. One of the most devastating multi-host viruses is the human immunodeficiency virus, or HIV. HIV is an RNA virus, and it attacks directly the helper T cells. So important components of our defense against viral infection is disarmed by the HIV virus. HIV exists in humans, but there are two different strains. The first strain, called HIV-1, seems to have come into humans from chimpanzees. The second strain, HIV-2, came to humans from mangabeys. In both cases, these were because of bushmeat consumption. In many parts of Africa, people eat monkeys, they eat apes. And these are viruses that would have gotten into the human bloodstream when somebody who just shot a monkey and was skinning it might have accidentally cut themselves with the same knife. We know that these viruses have come into humans hundreds of times, but only these two strains of HIV-1 and HIV-2 have the ability to transmit themselves from one human victim to another. Let's look now in detail at how HIV actually works. The virus is looking for a receptor along the cell wall. It finds one, integrates its own cell wall with that of the host cell, and then its RNA comes inside the host cell. Now, HIV carries along with its RNA a very important enzyme called reverse transcriptase. And the reverse transcriptase, once the RNA is inside the host cell, will now go backwards and make DNA from the RNA. Then the HIV's DNA strands now are integrated into the host DNA. And with HIV, if you're infected for life, because the virus spends most of its time inside of our chromosomes. Periodically, the chromosomes manufacture the RNA that will now code for the protein coat for the HIV virus, and all the bits and pieces necessary to reassemble HIV viral particles from within the host cell. Once these are reassembled, they then alter the surface of the host cell in a way that helps them to acquire now a protective cover as they leave the host cell and go on to infect new cells in the host body. So here they are getting ready to leave. And the life cycle of the virus is now complete. But notice that the host cell is still infected. It still carries then the DNA in there for the repeated manufacture of yet more and more RNA virus. Even though the viral particles have now left the host cell, the DNA from the virus is still there, and so it continues to manufacture more and more viral particles. Now, HIV is a particularly deadly virus because, first, it selectively attacks those helper T cells that are such a vital component of the immune system. So we saw how when there's a pathogen that comes into the immune system, there are these particular helper T cells that work with the macrophages and also with the B cells in order to mount an effective immune response. But these have been cut out, so they no longer can fight viral infection. The second thing about HIV is, unlike smallpox, the mechanism of inheritance in HIV is actually the RNA, because that's what travels in the particle from one host cell to another. And RNA viruses mutate much more rapidly than DNA viruses do. So here is an indication just of the speed with which HIV can mutate within a host. So this is a sequence divergence. All these different endpoints are the differences in the overall RNA of all the different strains now swarming within the same infected host who's only been infected for a few years. So these are 9 different patients. So this swarm is for this patient, this swarm is for this patient, this swarm from this patient, but a lot of divergence just within 6 years of an initial infection. So this presents a moving target for the immune system. What antibodies can be manufactured beside those that are being blocked by the action of the HIV against the helper T cells, they're chasing a moving target, and they don't necessarily recognize all viral particles and eliminate them within the body. The third virus I want to discuss today is influenza. This is the virus that causes a respiratory disease in humans, horses, pigs, and birds. Like HIV, influenza is a RNA virus. It's a single-stranded RNA virus. And many times we might think of influenza as just being like having a very serious cold. It makes us really uncomfortable. We're achy for a few days. But every now and then, influenza can be devastating to the point that there are quarantines, people were restricted in their movements. And in 1918, there was the most devastating disease outbreak, probably in short-term disease outbreak in human history. The Spanish flu of 1918 and 19 was one of the most devastating epidemics in human history. It struck Europe and North America right at the end of World War I. It actually was the reason that the war ended when it did, because so many troops fell ill. Worldwide, the disease killed between 50 and 100 million people in just 2 years. What was very frightening about this particular disease outbreak was the way that it killed young adult as opposed to younger or older people. Now, in normal circumstances, we expect babies to have higher mortality. They're frail when they're born, things can go wrong. And very old people, of course, are much more frail than young people. So typically, age-specific death rates look something like this, kind of a J curve, or a fishhook curve as it's called, with very low mortality rates during the primes of our life. But during the Spanish flu outbreak, the people that were disproportionately hurt were healthy young adults between the ages of 15 and 30, far higher than normal disease and mortality rate. Now, this had consequences on life expectancy. So as life expectancy has gradually increased with improved sanitation and nutrition over the last few centuries, In this overall climb, there is a remarkable drop in life expectancy coincident with the Spanish flu. So, what's the deal with Spanish flu and influenza in general? Influenza is an RNA virus, and every year you might be reminded, or you definitely will be reminded, to get your flu shots. And you might hear in the press or the words about what kind of flu there is this particular year. And you'll often hear like it's an H1N3 flu strain. And this is because there are two surface proteins on the flu virus that are very important for the way it enters into the host cells. And one of them is called hemagglutinin, and there's 16 different variants of that hemagglutinin protein. And that's where the flu actually attaches to the host cell. And then there's these neuraminidase proteins that release the viral particles into the cells. Now, you don't need to know these actual names, but it just might be interesting to keep in mind that when you hear about H1N3, for example, it's got a particular protein that gets into the cell and another protein that releases it. And that varies from year to year. Because these proteins vary so much from one year to the next, that's why we're carefully needing to have new vaccines. Now, these RNA viruses in the influenza, it's not one long strand of RNA, but it's actually a bunch of separate segments. And so we have then the name of the strain according to which protein of hemagglutinin and neurodine, so that's H and N. But what's important about this is that these RNA fragments, when they get inside the host cell, they're all mingled around, okay? And so the virus attaches itself using that H protein, it integrates into the host cell, it releases those RNA fragments. The RNA fragments start replicating themselves, as these viruses always do inside of a host cell. But it's an RNA virus again, so you get a lot of mutation in there, okay? And then when the virus is ready to leave, it grabs a selection of these RNA fragments and it goes off and it's ready to go. Now, the thing about these fragments is that every now and then it happens that some infected host is infected with two very different strains of influenza. And it might have gotten one strain of influenza from hanging around with its pigs or its ducks, and the other he might have gotten from somebody in the family. So if you've got these fragments from duck flu mixing in with fragments of human flu, they each would come in with their usual partners, but they might leave with a whole new combination of fragments that have never existed before in nature. So a flu virus may have exchanged genes with viruses from very different hosts and goes off and it's a brand new, weird, different kind of virus never seen before. So you have this rapid mutation inherent in RNA, and then you have the genetic mixing of different fragments coming together. It's a kind of recombination of different fragments from different viral strains. So you have a lot of reservoirs. You have reservoirs in ducks and birds, and they might be coincidentally infected with a number of different strains. You can have the virus circulating in pigs, other kinds of birds, even other kinds of mammals, horses, even whales and seals get influenza, as do mink. So all of these, if there's commingling and strange combinations going on, you can have, say, exposure to from your cat to you, as well as the flu you might get from another member of your own family. And horses and dogs can exchange it. So you get these wild, strange new combinations of flu. So what we've seen in history has been emergence of entirely new strains of influenza that are so different from each other that vaccines, of course, couldn't work because they're not recognizing these entirely new kinds of proteins that are appearing. So the flu genome codes a total of 10 different proteins, and it's on these 8 different RNA fragments. But each strain of flu may have those 8 RNA fragments originating from a lot of different hosts because they're being swapped in those co-infected hosts. So the Spanish flu is known as an H1N1 strain. It was the very first one that was characterized in great detail. so it gives the honor of H1 and N1 to those particular forms. And it went in from birds into humans. And this was known because you can actually, it has happened that scientists have gone to places like Norway, Siberia, and Alaska to graveyards where victims of the Spanish flu have been frozen ever since they died, and it's been possible to extract the virus and sequence it. All 8 of these segments from the Spanish flu in 1918 did come from a bird strain. Now, the bird strain may have originated from a lot of different hosts, but it was in birds for some time before it got into humans. Now, through time, there have been new strains, not nearly as devastating as Spanish flu, but there was a pretty serious flu outbreak around the world in 1957. This is called the Asian flu because the first cases were seen in Asia. And of the 8 different fragments in the flu there, 3 originated from birds and 5 had been circulating in humans. The next strain was the Hong Kong flu in 1968. You had 2 from birds, 5 from humans, and 1 from pigs. So in 2009, there was a lot of concern because there was another H1N1 strain floating around in various different parts of the world. And this H1N1 had those characteristic proteins of hemagglutinin and the other one, H1 and N1. They had obtained, though, their RNA fragments from 1 from humans, 2 from birds, and 5 from pigs. Now, the point of these is that these are each one so ridiculously different from the strain before that your vaccine, your memory cells aren't going to do you any good. There's such a different target now that's appearing each year from the new strain of influenza that you have to have a new shot every year. Now, the shot is much better than doing nothing, and the shot often works. It's not 100% successful, however, because the public health community has to guess which strain of flu will actually circulate in North America each year, and they have to concoct the vaccine several months before flu season actually begins. So it's a guessing game. They do their best to anticipate what fragments might be there, produce a suitable vaccine, and most of the time they're right. So it's definitely worth getting a flu shot every year, but every now and then they're not quite right, and you might still have a small risk of getting the flu. So what I wanted to show you with these 3 viruses is how our ability to live with them depends on the nature of the virus itself. So with smallpox, victims would be exposed only once, and there were 2 possible outcomes. Either you died of smallpox, or you survived, but you had lifetime immunity. And you had lifetime immunity because it was a stable DNA virus, it didn't mutate that much, and having the antibodies to your initial strain would help you against future infections. So vaccine was effective because of cross-immunity with whatever slight variants might appear in the future. HIV, on the other hand, is a virus that integrates permanently into the host genome. It never goes away. And it's all the time attacking the immune system. And it's an RNA virus that evolves very rapidly within the host. So you have an impaired immune system trying to track a moving target. Finally, we saw influenza or flu. It's another RNA virus. And besides having that rapid mutation rate, you have very complex patterns of recombination. And because these recombinants are so hugely different from each other, prior exposure are not effective against most novel strains because they're constantly emerging from many other host species. We've seen 3 major types of selfish genetic elements. First, we saw meiotic drivers. We saw how the Y chromosome, the Egyptian mosquito, the mitochondria of ladybugs distort sex ratios in their favor. We then saw transposons. These are stretches of DNA that can spread across chromosomes within the same individual and sometimes even spread between individuals. They can sometimes even be infectious. And then finally we saw the viruses. These are essentially the same as a transposon. They're selfish genes, but they have their own phenotype. And that phenotype is essential for being able to infect new hosts. Now, in all of these cases, we saw that there are defense mechanisms against selfish genetic elements. So with the meiotic drivers, we saw there were suppressor genes, which in the Egyptian mosquito was on the X chromosome to prevent the damage caused by the Y chromosome. Then repressor genes stop the transposons from jumping around within the genome. And we saw that within Drosophila. Finally, in viruses, we see that bacteria are able to cope with viral infection by restriction enzymes that attack specific sequences of DNA and basically obliterate it. And in our own immune systems, we have a number of different mechanisms that attack these viral antigens.